Chinese hamster ovary, or CHO cells, are commonly relied on for production of therapeutic proteins and antibody production, as they produce complex proteins comparable to antibodies that humans can produce. The metabolic characteristics of these cells are not widely recognized. Optimization of CHO cell culture relies on the information from hybridoma cell cultures in which both glucose and glutamine are energy substrates. Cell death occurs after glutamine depletion, and cell viability can be maintained with no glucose if glutamine is available, suggesting that glutamine can substitute for glucose. This study seeks to determine the effects of glucose and L-glutamine on growth and maintenance of recombinant CHO cells. The biological question being asked is, what is the significance of glucose and glutamine for CHO cell growth and maintenance? There is no explicitly stated hypothesis. However, it seems that the researchers think that both glutamine and glucose are necessary for growth and maintenance.
For assays and methods, we begin with a cell culture. Our CHO cells were subcultured in 75 milliliter T flasks after withdrawal from the cell bank. When growing to confluence, cells were dissociated with 0.025% tiosporin solution and pipetted into a single cell suspension as cell uniculum. 60 milliliter medium was put into 5 100 milliliter bottles, each bottle corresponding to a glucose concentration. Cell inicula were equivalently added to each of these bottles. The cell suspension in each bottle was distributed to 12 75 milliliter T flasks at 5 milliliters suspension per flask. The cell density in the bottles was the seeding density of these batch cultures. Batch cultures are displayed in the second picture on my slide. All T flasks were incubated at 37 degrees Celsius in a 5% CO2 incubator, and sampling was carried out twice a day. This process was used to display the growth of RCHO cells in batch cultures with different initial glucose concentrations. Next, batch cultures of RCHO cells at different initial glutamine concentrations were performed at low and high initial glucose levels. The experimental procedure was similar to that of the experiment at different initial glucose concentrations. This process was used to display the growth of RCHO cells in batch cultures with different initial glutamine concentrations. Cells were cultivated in a 500 milliliter spinner bottle with 150 milliliter medium containing 5 grams of microcarrier. The bottle was agitated at 60 RPM and the samples were taken twice a day. This was used with low glutamine concentrations and was done to compare against the original initial glutamine concentrations from the previous experiment. Viable cells were then counted by type-in blue exclusion using a hymaximeter. Every sample was counted 3 times and an average cell density was calculated. The concentrations of glucose and ammonia were determined using their respective assay kits. Lactate concentration was determined by a lactate dehydrogenase method. Amino acids were determined by reverse phase HPLC with pre-column deviation. Viable cells were counted to see if the density increased or decreased with different inductions of glutamine and glucose.
Our results showed that when the initial glucose concentrations were manipulated to levels between 8.9 and 49.6 millimoles, which can be seen in figure 1, maximum cell density did not change. However, when the glutamine concentration was manipulated so that there was 0.35 millimoles and a glucose concentration of 19.0 millimoles, cell density was lower, which can be seen in figure 3. The cell density was only 4.8 times 10 to the negative 5 cells per milliliter, which is far less than the 12 times 10 to the negative 5 cells per milliliter, which was seen at a higher glutamine concentration of 2.1 millimoles. However, viability stayed above 90%, as seen in figure 4 throughout this experiment. Despite the low glutamine concentration, the data showed that growth was exponential between hours 24 and 48. Then at 48 hours, cell growth slowed until hour 60 when it reached a steady level of cell density, which can be seen in the graph of figure 4. Based on reviewing the data in the graphs shown, I would have to agree with the author's interpretations of the results. The results showed that despite hybridoma cell research showing glutamine as having the potential to substitute for glucose, this is not the case for our COH cells. The results show that glutamine is pivotal to cell respiration in RCHO cells. Additionally, although glutamine may be necessary for proliferation, it is supported by the experimental data that in terms of cell maintenance, glutamine is not necessary. Glucose can uphold cell maintenance without glutamine. So, in sum, this makes sense when corresponding with the data because the data showed cells being necessary for proliferation and growth, but not for maintenance of the cell. Hence why I agree with the author.
With this experiment, we could see that the conclusion was definitely made clear in the piece. It states that in RCHO cell culture, glucose was an absolute needed nutrient for the cell growth and the cell maintenance. So it was needed for the cell to grow, and it was needed for the cell to survive and just maintain itself without growth or depletion. It also states that, on the other hand, glutamine was essential, but glutamine was not essential for cell maintenance. It was essential for cell growth. It also states that RCHO cells could neither grow nor remain viable. So they couldn't grow and they couldn't just maintain on its own without being grown or depleted in the absence of glucose. And glutamine could not substitute for glucose as the sole energy or carbon substrate. So basically what this means is they came to the conclusion, as stated in the introduction, an abstract, that glutamine was not something that could stand in the place of glucose for cell growth and cell maintenance. So if you are without glucose, then the cell will definitely die. But if you are without glutamine, the cell can still maintain itself and grow if and only if there was glucose present. Therefore, we do agree with their conclusion because in the results, you could see that they stated very clearly that without the glucose, these cells began to deplete and they began to die, or there was no growth. And they also made clear figures, or the illustrations that are in the piece, which also gave us a very easy to read image of what was actually happening. So therefore we can conclude that we agree with the conclusion. As far as ways that this experiment could help in the future, in the introduction it stated that not much is known about the energy metabolism and regulation modes in CHO cell culture processes. And since they don't know much about it, they have to use the hybridoma cell cultures. And with this experiment, they proved that you need only glucose in the CHO cells as the energy source, but in hybridoma, you needed both. So they can see the difference in the energy needs and the metabolic pathways of these different cells. And now, with the conclusion of this experiment, they can see that, or they found that the CHO cells only need glucose, therefore they don't have to really depend on the hybridoma cell cultures. So in the future, they can continue to to process these CHO cell cultures, because these CHO cell cultures are actually useful in experiments that produce therapeutic proteins such as prothrombin and other blood clotting features, features of proteins. So basically, this experiment really helped to target the CHO cells as the primary source of the prothrombin experiments or the production of therapeutic proteins.